Welcome back to the Deep Dive. We're here to unpack complex topics and give you the essential insights. Today, we're really venturing deep into genetics,
Mm.
but maybe not the kind you first think of. We often think of inherited traits as pretty clear cut, right? Like your blood type A, B, ABO. Simple categories.
Or maybe fruit fly eye color, red or white,
Yeah,
very distinct.
exactly. But here's the really fascinating thing. Most traits, the vast majority in all living things, they aren't quite so simple.
That's absolutely right. Think about something like human height, or say how much grain a particular wheat plant yields.
Or even, your susceptibility to certain diseases.
Right, these aren't just either situations, they exist on a continuum, a whole
So-
spectrum. There's just endless variation. You measure them, you don't just sort them into boxes.
And that continuous spectrum, that's exactly what this deep dive is about today. We're talking quantitative genetics and multifactorial traits.
Yep.
Our mission really is to explore how these incredibly complex characteristics influenced by, wow, many genes plus a whole bunch of environmental factors, how we can still understand them. We can actually use Mendelian principles, but like boosted with some powerful statistical tools.
Exactly. We're going to look at how geneticists estimate the genetic part of a trait, how they cleverly use twin studies to figure things out,
Yeah.
and even how they can actually pinpoint specific genes involved in these really intricate processes.
So by the end of this, you should have a really solid grasp of a critical area in genetics, and hopefully some surprising facts, some real-world applications that might just change how you view things a little.
Definitely.
Okay, so let's start there. We're talking about traits that vary continuously, unlike, say, Mendel's peas, which were just tall or short, green or yellow. Can we maybe clarify some key terms first? What exactly is continuous variation?
Yeah, good place to start. Continuous variation just means traits you can measure quantitatively. Think weight, height, crop yield. You don't get neat categories, you get a whole range of values.
Okay, a measurable range.
And that naturally leads to polygenic traits, literally, of many genes. It means the different phenotypes, the different versions we see, result from the combined input of genes at more than one place, often many places, on the chromosomes. We call those individual genes polygenes.
Right, so polygenic means many genes contribute to one trait outcome. But you mentioned another layer of complexity, something that makes them complex.
Precisely. That brings us to multifactorial traits, sometimes called complex traits. These are quantitative polygenic traits where the final phenotype, what you actually see or measure, is influenced by both the genes and environmental factors.
The nature and nurture combination.
Exactly. Human height is the classic example. Your genes sort of set a potential range, but your nutrition, your health during childhood, other environmental things, they play a huge part in where you actually end up on that scale.
So genes load the gun, environment pulls the trigger, kind of.
That's one way to think about it, yeah. And just for clarity today, we'll stick with the term multifactorial.
Okay, multifactorial. It still feels like a pretty big leap from Mendel's simple pea plants, discrete traits, to something as messy as human height. How did the early geneticists even connect continuous variation back to Mendel?
That's a great question. And it was a huge point of debate back in the early 1900s. Some scientists really argued that Mendel's unit factors, what we now call genes, could only explain those clear-cut, discontinuous traits.
They couldn't see how it applied to a smooth range.
Right. They struggled to see how something like height, with all its tiny differences between people, could be explained by distinct, separate units of inheritance.
So how did they bridge that gap? What was the insight?
Well, the breakthrough really came from William Bateson and Nilsson-Ehle. They proposed what they called the multiple factor or multiple gene hypothesis.
Okay.
Their idea was that maybe many genes, each one following Mendelian rules individually, could contribute to a single phenotype in a sort of cumulative quantitative way, additive effects.
Additive effects. And wasn't there a key experiment with wheat that really nailed this down?
There was. Herman Nilsson-Ehle's work in 1909 with wheat grain color. It was absolutely pivotal. He crossed a variety with dark red grain to one with white grain.
And the first generation, the F1.
They were all intermediate pink. Now, your first thought might be, okay, incomplete dominance, like snapdragon flowers. One gene involved.
Makes sense. But the next generation, the F2, that's where it got interesting.
That's where it got really interesting. He didn't get the simple 3.1 or 1.2.1 ratios you'd expect from a single gene. Instead, he saw a whole range of colors, from dark red all the way back to white. And crucially, they appeared in a specific ratio, about 1 dark red, 4 medium dark red, 6 intermediate pink, 4 light pink and 1 white.
A 1.4.6.4.1 ratio, that adds up to 16.
Exactly. That ratio, in 16ths, strongly suggested that two different genes, each with 2 alleles, were controlling the color, and they were segregating independently, just like Mendel predicted for 2 unlinked genes.
So the different shades weren't just about one gene being on or off, but how many red alleles you got in total.
Precisely his insight. He proposed each of the 2 genes had 1 additive allele, let's say it contributes 1 dose of red pigment, and 1 non-additive allele that contributes no pigment.
Okay.
So the dark red parent might be, say, AABB with 4 additive alleles. The white parent AUB, 0 additive alleles. The F1 hybrid would be ABAB with 2 additive alleles, hence intermediate pink.
And in the F2, you get all the combinations.
All the combinations from AABB, 4 additive alleles, dark red, down to AAB, 0 additive alleles, white, and everything in between, 3 doses, 2 doses, 1 dose. The more additive alleles, the darker the red.
Wow. So that single experiment elegantly showed how continuous variation could arise from discrete Mendelian genes acting together. It wasn't defying Mendel. It was Mendel, just more of him working together.
It really was a critical aha moment. It showed that these complex quantitative traits could be explained by Mendelian inheritance with multiple genes, each contributing a small additive amount to the final phenotype.
Okay, so if these traits are controlled by lots of genes, each chipping in a bit, and you add environmental effects, you obviously can't just count simple ratios like 3.1 anymore.
Definitely not.
This is where statistics has to come in, right?
Absolutely.
Yeah.
That's where the power of numbers becomes essential. Because you have this continuous variation, those classic Mendelian ratios just don't apply directly. Geneticists have to rely heavily on statistical analysis, and they study whole populations, not just individual families.
Right. You need the big picture, which means you need good data from a lot of individuals. I imagine getting a reliable snapshot of a whole population is tricky.
It really is. And the importance of proper sampling just can't be overstated. You need a sample that's large enough, chosen randomly, and actually represents the population you're interested in.
Like, you wouldn't measure the average height of, say, university students by only measuring the basketball team, right?
Exactly. You'd get a very, very skewed result. But when you do have a large representative sample, the data for a quantitative trait very often forms a normal distribution.
Ah, the bell curve.
The famous bell-shaped curve, yeah. When you plot the frequency of each measurement, most individuals cluster around the average, with fewer and fewer at the extremes.
Okay, the bell curve. So, what statistical tools do geneticists use to actually analyze that data, to describe that curve?
Several key tools are indispensable. First, you need the mean, which is just the arithmetic average. It tells you the central point of your measurements.
The peak of the bell curve, roughly.
Kinda, yeah. But then you need to know how spread out the data is around that mean. How wide is the bell? That's where variance comes in. It quantifies the spread. Technically, it's the average squared distance of all measurements from the mean.
Square distance. Okay, so a higher variance means a wider, flatter curve. Less variance, a taller, skinnier one.
Precisely. But variance is in squared units, which can be a bit awkward to interpret. So we often use the standard deviation, which is just the square root of the variance.
Okay. Back to the original units.
Right. And the standard deviation is really useful because for a normal distribution, about 68% of all the values fall within 1 standard deviation of the mean.
Okay.
And over 95% fall within 2 standard deviations. So it gives you a really good handle on the spread and probability within your data set.
Got it. Mean for the center, standard deviation for the spread. What if you're interested in how 2 different traits vary together? Like, do taller people tend to weigh more?
For that, you'd look at covariance. It measures how much variation is shared between 2 different quantitative traits.
Okay.
And to make it easier to compare across different studies or traits, we often standardize the covariance into the correlation coefficient, usually denoted as R.
The R value. I've heard of that.
Yeah, it ranges from -1 to +1. A positive R means as one trait increases, the other tends to increase as well, like height and weight, probably. A negative R means they tend to move in opposite directions. 0 means no linear association.
So positive R for height and weight, maybe a negative R for time spent studying and number of TV shows watched?
Could be. But here's the absolutely critical point, and it bears repeating. Correlation does not prove causation.
Right. Just because 2 things happen together doesn't mean one causes the other.
Exactly. There could be a third factor influencing both, Or it could be coincidence. Correlation just tells you there's an association in the variation patterns. It doesn't tell you why.
That's such an important caveat. Okay, let's make this more concrete. The source material used a tomato fruit weight example, a cross between an 18-ounce variety and a 6-ounce one.
Right. The F1 generation, the hybrids, had fruits mostly between 10 and 14 ounces intermediate, as you might expect.
But the F2 generation, that's where the variation exploded.
It did. The F2 plants produced tomatoes across the entire range, from 6 ounces all the way up to 18 ounces, just like the original parents. The average weight was actually similar to the F1, around 12 ounces.
But the spread was way bigger.
Way bigger. The variance calculated for the F1 was about 1.3, but for the F2, it jumped to over 4.2. The standard deviation also nearly doubled. It clearly shows that segregation of multiple genes in the F2 generation leads to much greater phenotypic diversity.
And you can even use these stats to estimate how many genes are involved. That seems amazing.
Yeah, there are ways to estimate it. For instance, you can look at the proportion of F2 individuals that resemble one of the extreme original parents, using formulas like 1 over 4 to the power of n, where n is the number of genes.
Whoa, okay, maybe a bit technical here, but the principle is you can estimate n.
Exactly. In that tomato example, if maybe 1 out of 72 F2 plants produce tomatoes like the original small or large parents, the formula suggests that probably 3 or 4 genes are contributing most significantly to that fruit weight difference.
3 or 4 genes. Okay. So we know these traits are complex, influenced by many genes, influenced by environment. But the big question, the one that always comes up, how much is nature, how much is nurture? How do geneticists actually quantify that contribution?
Right, that brings us squarely to the concept of heritability. In genetics, heritability describes the proportion of the total phenotypic variation you see in a population that is due to genetic factors.
So the variation, not the trait itself.
Critically, yes. It's about what proportion of the differences between individuals in a population can be attributed to differences in their genes under the specific environmental conditions they experienced.
A high heritability means genetics explains a lot of the variation.
Correct. A high estimate suggests that much of the observed variation is due to genetic differences among individuals, with the environment playing a smaller role in creating those differences. A low estimate suggests environmental factors are more responsible for the variation seen.
No, this is where people often get tripped up, right? It's probably the biggest misconception. Heritability is not about how much of your trait is genetic.
Absolutely critical distinction. A heritability estimate of, say, 0.65 for human height doesn't mean 65% of your specific height comes from your genes and 35% from your environment. That makes no sense for an individual.
It's about the population.
It's about the variation within a specific population living in a specific range of environments. It's a population measure, not an individual one. And it can change if the environment changes.
That's such a crucial point, like the chicken-egg example you mentioned.
Yeah, perfect example. If you have a flock of chickens all kept in identical cages with identical food, any differences in egg production are likely due to their genetic differences. Heritability would be high.
Makes sense.
But take that same flock and let them range freely outdoors. Now, variation in egg laying might be due to who's better at finding food, who gets a better nesting spot, who avoids predators. Environmental factors become much more significant, and the heritability estimate for egg production in that environment would be much lower.
Same chickens, different environment, different heritability.
Exactly. So heritability doesn't imply genetic destiny
Yeah.
or that a trait can't be changed by the environment. It's always relative to the population and environment studied.
Okay, that's crystal clear. So how do geneticists actually calculate these estimates? How do they break down the variation?
They do it by partitioning the total phenotypic variance. That's the variation we actually observed. Symbol VP into different components. The basic equation is VP equals VG plus VE plus VGXE.
Okay, VP equals VG plus VE plus VGXE. Let's break that down.
VP is the total phenotypic variance. VG is the portion of that variance caused by differences in genotypes among individuals, the genotypic variance. VE is the portion caused by differences in the environments individuals experienced, environmental variance.
And the last term, VGXE.
That's the genotype by environment interaction variance. And it's really important, often tricky. It accounts for cases where different genotypes respond differently to different environments.
So it's not just adding genes and environment, but how they interact specifically.
Precisely. Think of 2 different crop varieties. Variety A might yield okay in poor soil and only slightly better in good soil. Variety B might yield poorly in poor soil, but fantastically well in good soil.
So variety B responds more to the better environment.
Exactly. That differential response is VGXE. The effect of the environment depends on the genotype, and vice versa. It highlights why a simple nature versus nurture debate is usually way too simplistic. It's an interplay.
Okay, and within that genetic variance, VG, are there different types too? You mentioned different kinds of heritability.
Yes. The first type is broad sense heritability, written as h squared,
Hmm.
HVA. This measures the total contribution of genotypic variance, VG, to the total phenotypic variance, VP. So H versus VG, VP.
Broad sense includes everything genetic.
Everything genetic, yes.
Hmm.
Which is interesting, but it has limitations, especially for animal and plant breeders, because VG includes different kinds of genetic effects, some that are easily passed on, some that aren't.
Like dominance effects.
Exactly. VG can be further broken down into VA, additive variance, VD, dominance variance, and interactive or epistatic variance. Additive variance VA comes from the average effect of alleles added together. Dominance variance VD arises because one allele can mask another at the same locus. Interactive variance comes from interactions between different loci epistases.
And breeders care most about the additive part.
They care most about VA, the additive variance, because additive alleles contribute predictably and cumulatively to the phenotype, and they're the ones reliably passed from parent to offspring. Dominance and interaction effects depend on specific combinations of alleles, which get broken up during reproduction.
That makes sense. So there's a heritability measure just for that additive part.
Yes, and that's narrow sense heritability, written as h squared.
Right.
It's the proportion of the total phenotypic variance that is due only to additive genetic variance. So HY equals a VAVP.
And this is the one that's really useful for predicting response to selection.
This is the key one for breeders. Narrow sense heritability tells you how much of the variation is breedable, how much progress you can expect to make by choosing the best individuals to be parents for the next generation. This is the basis of artificial selection.
Artificial selection choosing the best to breed.
Right. The higher the narrow sense heritability for a trait, the more effective selection will be in changing the average phenotype of the population over generations. We can even measure realized heritability by looking at the actual response achieved after selection.
Like that amazing corn oil experiment in Illinois, that's been running forever.
An incredible example, yes. Started in 1896. They've been selecting corn for both high and low oil content for, well, over 100 generations now. I think the source said 76 were analyzed in detail.
And they saw huge changes.
Huge changes. In the line selected for high oil content, the average oil percentage increased dramatically over those generations, showing a strong response to selection and therefore significant additive genetic variance initially.
But did the heritability stay high?
Interestingly, no. As selection proceeded and the desirable alleles became more common, the amount of additive genetic variance remaining in the population decreased. So the narrow sense heritability actually declined over time, meaning further selection became less effective as they approached the genetic limits.
That makes perfect sense. You use up the variation you're selecting for. It also sort of explains why traits really essential for survival often have lower heritability, right?
Exactly. Things like fertility or survival rate have likely been under intense natural selection for millennia. Natural selection has already favored the optimal alleles and reduced the genetic variation for those traits. There's less additive variance left for artificial selection to work with. Traits less critical for survival often retain more genetic variation and thus have higher heritability.
Okay. But we always have to remember the limits, right? Heritability doesn't tell you which genes are involved.
Correct. It tells you nothing about the number or identity of the genes.
It only applies to the specific population and environment studied.
Absolutely. Change either, and the estimate might change.
And it depends on the environmental variation present. If there's no variation in environment, VE is 0, and H-light might seem artificially high.
A very important point. And it can't predict future changes if the environment shifts significantly.
Got it. Okay, so studying heritability in humans is obviously way trickier. No controlled breeding programs. This is where twin studies come in as such a powerful tool.
Immensely powerful, yes, because twins give us a sort of natural experiment. We have monozygotic, or MZ twins, identical twins. They originate from a single fertilized egg that splits, so they are traditionally considered genetically identical at conception.
One zygote, monozygotic,
Yeah.
identical.
Right. Then we have dizygotic, or DZ twins, fraternal twins. They result from two separate eggs being fertilized by two separate sperm. So they are genetically no more similar than any other pair of siblings, sharing on average 50% of their segregating alleles.
Two zygotes, dizygotic, like regular siblings, just born at the same time.
Essentially, yes, and the logic is since MZ twins share 100% of their genes, or very close to it, and
Okay.
DZ twins share 50%, we can compare them to disentangled genetic and environmental influences.
How does that work? By looking at whether they share a trait.
Exactly. Researchers look at concordance rates. A twin pair is concordant if both twins express the trait. They're discordant if only one does.
Okay.
So the idea is if a trait is strongly influenced by genetics, you'd expect the concordance rate to be significantly higher for MZ twins than for DZ twins.
Makes sense. If genes are key, identical genes should lead to the same outcome more often.
Right. Think about traits like blood type or eye color. MZ concordance is virtually 100%, while DZ concordance is much lower, reflecting Mendelian inheritance patterns. This points to a very strong genetic determination.
But sometimes high concordance doesn't automatically mean high genetic influence, right? Like the measles example.
That's a crucial counterpoint. Measles used to show high concordance in both MZ and DZ twins. Why? Because it's caused by a virus and environmental factor. If both twins are exposed, both are likely to get sick, regardless of whether they're MZ or DZ.
So you need to look at the difference between the MZ and DZ rates.
Precisely. A substantially higher MZ concordance compared to DZ concordance is the key indicator of a significant genetic component to the variation in that trait.
Okay. But what's really mind-blowing is that recent discoveries are challenging the fundamental assumption that MZ twins are perfectly identical genetically.
It's been a fascinating refinement of our understanding. We now know MZ twins aren't always 100% identical genomically, even
Right.
from birth. One reason is copy number variations, or CNVs.
Copy number variations,
Yeah.
differences in chunks of DNA.
Yeah, relatively large segments of DNA that can be duplicated or deleted. These CNV events can actually occur after the zygote splits, but early in development. This means the two twins can end up with slightly different copy numbers for certain genes or regions.
Wow. So they start as one cell, but slight changes can happen as they divide.
Exactly. It leads to what's called somatic mosaicism, where different cells in the body might have slightly different genomes. And these CNV differences between MZ twins have been linked to discordance for certain conditions, like specific types of leukemia.
So that's one crack in the identical label. What else?
The other big area is epigenetics. While MZ twins have the same DNA sequence, their epigenome, the chemical modifications to the DNA and associated proteins that regulate gene expression can differ.
Like methylation?
DNA methylation is a key one, yes. Also, histone modifications. These epigenetic marks help determine which genes are turned on or off in which cells. And while MZ twins are epigenetically very similar at birth, their epigenetic patterns tend to diverge as they age, influenced by different environmental exposures and experiences.
So same genes, but different instructions on how to use them over time.
That's a good way to put it. And these epigenetic differences can lead to phenotypic discordance, even for traits with a strong genetic basis. Beckwith-Wiedemann syndrome, a developmental disorder, is a known example where discordant MZ twins show clear differences in methylation patterns.
So these discoveries, CNVs and epigenetics, they don't invalidate twin studies, do they? They just add another layer.
They refine them. They show that phenotypic differences between MZ twins aren't only due to the large-scale external environment, but can also arise from subtle internal genomic or epigenetic changes that occur post-conception or accumulate over life. It actually makes twin studies even more powerful for dissecting these complex interactions.
And didn't a huge study recently pull together data from tons of twin studies?
Yes, a landmark meta-analysis in 2015.
Yeah.
It was massive, looked at data from over 14 million twin pairs across something like 17,000 different human traits.
Wow. What did it find overall?
It strongly supported the general importance of genetic factors across almost all traits studied. But critically, it also reinforced the idea that most complex traits are highly polygenic, influenced by many genes, each having only a small additive effect. This aligns really well with what we see from other large-scale genomic studies like GWAS. They even created a web resource, MatTCH, if people want to explore the data.
Okay, so twin studies help estimate the overall genetic contribution. But how do we actually find the specific genes involved in these polygenic traits? That still seems like looking for needles in a genomic haystack.
It is definitely challenging, especially with all the environmental noise and gene interactions. But the main approach today involves identifying quantitative trait loci, or QTLs.
QTLs, what exactly are those?
A QTL is basically a specific region on a chromosome that contains one or more genes that contribute to the variation observed in a quantitative trait. It's a location, a locus.
So it's a neighborhood where a gene influencing the trait lives. How do they map these neighborhoods?
The typical strategy starts by crossing two parental lines that are very different, highly divergent. For the trait you're interested in may be created through long-term artificial selection.
Like the high and low oil corn lines.
Could be, yeah. Or different tomato varieties. You cross them to make an F1 generation, then cross the F1s, or self-fertilize them to create an F2 generation.
And the F2s are all mixed up genetically.
Exactly. Each F2 individual inherits a unique mosaic of chromosome segments from the original two parental lines. So you get a population with lots of variation both in the trait and in their underlying genomes.
Then you look for connections between the genome bits and the trait.
Precisely. You genotype all the F2 individuals using lots of DNA markers spread across the entire genome. These markers are like signposts. They could be RFLPs, microsatellites, or now most commonly SNPs, those single-letter DNA variations.
Okay, you've got marker data and trait data for everyone.
Then you use statistical analysis to see if any specific marker genotypes are associated with variations in the quantitative trait. If a marker is physically close on the chromosome to a gene that influences the trait, QTL, then the marker and the QTL gene will tend to be inherited together. They'll co-segregate.
So individuals with marker version A might tend to have higher trait values than individuals with marker version B.
Exactly. If you see that kind of statistically significant association, it suggests that a QTL influencing your trait is located near that marker on the chromosome. You get a peak on a graph showing the likelihood of a QTL at different map positions.
That's clever. And the tomato example, again, the size difference is huge.
It's dramatic. Wild tomatoes weigh maybe a few grams. Cultivated ones can top 1,000 grams. That's like a 1,000-fold difference. Research, particularly by Stephen Tanksley's group, identified numerous QTLs involved.
And they even found specific genes within those QTLs.
They did. One major QTL called FW242, located on chromosome 2, was found to contain a gene called ORFX. Variations in this single gene can account for up to 30% of the fruit weight variation between wild and cultivated tomatoes.
30% from one gene, what does it do?
It seemed to act as a negative regulator of cell division. So the cultivated varieties have versions that lead to lower expression of this gene early in fruit development, allowing more cell division and ultimately much larger fruit.
Fascinating. And weren't there other QTLs for things like the number of compartments inside the tomato?
Yes, the locules, the seed compartments, QTLs like LC and FAST control locule number. Ancient tomatoes had maybe 2 or 3. Modern beefsteaks can have 10 or more, which also contributes to size and shape. And these QTLs can interact with each other too.
So QTL mapping isn't just theoretical. It's led to identifying specific genes with major effects in crops.
Absolutely. It's been hugely important in plant breeding for corn, rice, wheat, you name it, and also in livestock breeding for traits like milk production or meat quality.
Now, there's another related concept, eQTLs, expression QTLs.
Right. So QTLs are loci affecting the trait itself. eQTLs are genomic loci that regulate the expression levels of other genes.
So they control how much product other genes make.
Essentially, yes. An eQTL might contain a non-coding variant, like an SNP, that affects how efficiently a gene somewhere else is transcribed, maybe by altering a promoter or enhancer region, or affecting RNA processing.
And these eQTLs are important for understanding human diseases.
Very much so. Many genetic variants associated with complex diseases through genome-wide association studies, GWAS, actually turn out to be eQTLs. They don't change a protein structure, but they change how much of it gets made, or where, or when.
Can you give an example?
Asthma is a good one. Researchers integrated GWAS data for asthma risk with eQTL data from relevant lung tissues. This helped them move beyond just statistical associations to pinpoint actual genes whose expression levels were linked to asthma risk. They identified networks of interacting genes, including potential driver genes that regulate others in the network.
Driver genes, so potential drug targets.
Exactly. By understanding how genetic variation affects gene expression networks involved in disease, we can identify much better targets for developing new therapies.
Looking back at the bigger picture, the real-world impact of understanding quantitative genetics seems enormous. What stands out most?
Well, you absolutely have to mention the first green revolution. In the 1950s and 60s, applying principles of quantitative genetics and breeding led to massive increases in the production of staple crops like rice, wheat, and maize.
Feeding a growing world population.
It was critical. The International Rice Research Institute, IRI, played a huge role. They developed varieties like IR8, which was high-yielding and disease-resistant.
But it had that lodging problem, right? Too heavy on top.
It did, yeah. It produced so much grain it would fall over before harvest. So the breeders deliberately crossed IR8 with shorter, dwarf varieties. The resulting semi-dwarf lines had strong stalks that could support the heavy grain heads.
And that combination was revolutionary.
It truly was. Those semi-dwarf high-yield varieties helped double world rice production in about 25 years, averting widespread famine in many parts of Asia. It's a stunning success story for applied quantitative genetics.
But the challenges haven't stopped. We hear about needing a second green revolution.
We absolutely do. The global population continues to grow, and we face new challenges like climate change, requiring crops with even higher yields, but also better tolerance to drought, heat, salinity, and new pests and diseases. Quantitative genetics, QTL mapping, genomic selection, these are all crucial tools for meeting those future food security needs.
It really underscores the ongoing importance of this field. Now, this deep dive into complexity also touches on some, well, profound ethical considerations, especially with modern tech like whole genome sequencing.
It does. When you sequence someone's entire genome, perhaps looking for the cause of one specific condition, you might unintentionally uncover information about their risk for other unrelated conditions, like finding variants associated with a higher risk for Alzheimer's disease later in life.
And that raises really difficult questions.
Huge questions. What information should be disclosed? To whom? How do you communicate that risk information responsibly, especially when it might be uncertain or impact family members? Genetic counselors grapple with these complex ethical issues constantly. It highlights how powerful this knowledge is and the responsibility that comes with it.
It's a lot to think about. We've certainly covered a huge amount of ground today, from those simple Mendelian traits to this incredibly intricate dance between, dozens or hundreds of genes and countless environmental factors.
It's amazing how far we've come. Using statistical tools, leveraging twin studies, mapping QTLs, we really are starting to unlock the secrets of these complex biological puzzles. Our understanding of life's blueprint just keeps getting deeper and more nuanced.
It really does. So as we wrap up this deep dive, maybe a final thought for you to ponder. Given this incredible complexity and the new discoveries constantly challenging even what we thought was solid, like MZ twin identity, how might our understanding of health, of medicine, even of our own identities continue to shift in the coming decades?
Yeah, what does genetic predisposition really mean when the interplay is this complex? It's something to keep thinking about.
Definitely. And maybe take a moment in your daily life to notice the quantitative traits all around you, the variation in height and plant growth, maybe even in your pets.
Thank you for joining us on this exploration of quantitative genetics. It's been a fascinating journey.
It has indeed. Thanks for being here for the deep dive.